Please, let's be quiet, please. Family Enterobacteriaceae. The Enterobacteriaceae are a large diverse family of gram-negative, facultatively anaerobic, rod-shaped bacteria, commonly found in the intestinal tract of humans, and animals. Commonly found in the intestinal tract of humans and animals. Commonly found in the intestinal tract of humans and animals, as well as in soil, water, and plants. Where is it commonly found? Intestinal tract of human. It can also be found in the soil, but because it is intestinal in origin, if you now see it, it's intestinal in origin. If you now want to drink water, you now see it inside water, you'll be assuming that if this thing that is from intestine can get to water, that means that fecal matter, excreta, because what is in your intestine will get to the water. How? They don't open somebody's intestine, it will go through excreta. You will now figure that, that means this water contains excreta, because members of Enterobacteriaceae have been found in that water or in that food. So what do? You take the food and enjoy it, Abi? You put it aside. You do what? You put it aside. You get to that when you get to water and sewage microbiology in 400 level. It's very key. This family is very, very key family. They are among the most medically significant families. They are among the most medically significant bacterial families. They are among the most medically significant bacterial families containing. Where is the assistant class rep? Assistant class rep, where are you? Class rep, where are you? No class rep, no assistant. Are you the assistant class rep? Containing both pathogenic and opportunistic species. Who want to stand in for the class rep? Who want to stand in for the assistant class rep? Okay, those two boys standing there. You are now the class rep, you are now the assistant. Okay, what's the difference between pathogenic and opportunistic? Why are you laughing now? Why are you laughing? Why is the laughter? Please. I'm recording. Please don't spoil my recording. Even the people, they are ready to answer the question, and you people are laughing. The two of you there, what is the difference between pathogenic and opportunistic species? What is the difference between pathogenic and opportunistic species? What is the difference between pathogenic and opportunistic species? What is the difference?
Pathogenic species.
Speak out now, speak out. What you are, if it's,
Pathogenic species causes diseases, while opportunistic species, they don't necessarily cause disease.
So what do they cause? They cause health. Yes. What is the difference between pathogenic and opportunistic species? She said, and she's correct, that pathogenic species are species that cause disease, that bacteria that cause disease. So what are opportunistic species? What are opportunistic species? What? They benefit from the disease. Benefit from where? What are opportunistic species?
They also call only when the cell is susceptible.
They cause disease when what?
The cell is susceptible.
When?
The cell is susceptible.
Which cell? When conditions. When conditions are favorable. Pathogenic species cause disease. Once they land like this, they will cause disease. But opportunistic species, they can be on your skin. Like what organism is in your skin? Normally. Normal flora of your skin. Speak out now, far. Speak out. Don't be afraid. If you miss it, I'm not going to. There's nothing I will do. I will just look at it and move on. So, normal flora of your skin is what? Staphylococcus staphylococcus. Which organism is that one? No. Which organism is normal flora of your skin? Epidermidis. Even aureus. What about your mouth? Let's hear from this man that is covering his mouth. The normal flora of your mouth is what? The same staphylococcus. It's a normal flora of your mouth. And that is why the guy is using mask. So as he's talking, staphylococcus in his mouth does not come out. I'm not joking, no. I think he's covering his nose so that he will not inhale any staphylococcus from anybody. But staphylococcus is normal flora of the skin. Normally, it's on your skin. Whether you like it or not, it's on your skin. You get it now. If it's on your skin, it's on your cloth now, Abi. If it's on your cloth, it's on your chair now. If it's on this table now, you get it now. Like the two people that left this table now, they start on their hands, on their books. As I'm touching the table, is getting attached to my hand. But my hand does not have any wound. So nothing. Can't do anything to me. But the moment there's a cut, then the staphylococcus on the skin will get into the bloodstream and then cause infection. That's opportunity. The opportunity is the cut. So, there's cut. Normally it's on the skin. It will not enter. It will not enter on broken skin. But the moment the skin is broken, it is going to get in. And once it gets in, it's going to cause infection. That is opportunistic, pathogen. But on a normal day, it's not a pathogen like that. But when the opportunity comes, it causes pathogen. It causes infection or disease. That I said that normally it's not a pathogen. If Salmonella staphylococcus is found in another place, which is not a normal flora, it's a pathogen. But for the skin, it's a normal flora of the skin. So once the skin is not broken, it will not cause infection. You understand? Any organism that is normal flora of somewhere, like intestine now, there are some organisms that are normal flora of the intestine. Like what? Like what? Enterobacteriaceae. Enterobacteriaceae. Let's go on. Members of this family are straight rods. They are straight rods. Gram-negative, motile by peritracheous flagella. Motile by peritracheous flagella. You see, there's no board for me to write to. It's not my fault too. Motile by peritracheous flagella or non-motile. They are non-sporing. Some species possess capsule. Leave it. So go and sit down. Some species possess capsule. Some species possess capsule, e.g. Klebsiella. They are facultative anaerobes. They are facultative anaerobes. That is, they grow in both aerobic and anaerobic conditions. That is, they grow in both aerobic and anaerobic conditions. They are catalase positive. They are oxidase-negative. They are oxidase-negative. This is a key distinguishing feature. This is a key distinguishing feature from Pseudomonadaceae. This is a key distribution feature from Pseudomonadaceae. Please don't stay in that place. Go and stay somewhere else. Pseudomonadaceae. They reduce nitrate to nitrite. And take it off. That socket, is it not working? It's what? This is not no light on top of this thing. There's light on this thing. This one on the floor. This thing that is on the floor, plug it to that something. There's no socket. Eh? This wire that's on the floor. This one I want to plug. There's no extension. That light that's coming here, there's no light inside. They reduce nitrate to nitrite. Please, let's pay attention. They ferment glucose. They ferment glucose with acid and gas production. They ferment glucose with acid acid and gas production. The optimum temperature for growth is 37 degrees C. The optimum temperature for growth is 37 degrees C. Members of Enterobacteriaceae are normal flora of the intestinal tract of humans and animals. Members of Enterobacteriaceae are normal flora of the intestinal tract of humans and animals. They are found in soil, water, sewage, and on plants. They are found in soil, water, sewage, and on plants. Some are free living. Some are free living, while others are opportunistic. Some are free living, while others are opportunistic or primary pathogens. They are free living, while others are opportunistic or primary pathogens. So you can see, some they are in the intestine, some are in the soil, some are free living. But that does not mean, because they are intestinal in origin, does not mean that when we now see it in food, we now say it's free living, let's eat the food. We now see it in water, we now say it's free living. No, we assume it's intestinal in origin. So if it's intestinal in origin and we found E. coli in the water or in the food, we also assume that that water or food is polluted. You get it now? The last time I gave you an assignment that you find out the meaning of facultative anaerobes. Facultative aerobes. Did you find them? Yes. If you can define it, let me see your hand up. From this side, at the back there. Facultative anaerobes or facultative aerobes. If you can define it, raise your hand from the outside. Anybody from this side? Anybody from the back there? Anybody from this side? I gave you an assignment that you find out the meaning of facultative aerobes and facultative anaerobes. Did you find it? Yes. You found it? So what is the facultative anaerobes? She said facultative anaerobes, they can survive in both presence of air and absence of air. How far is she correct? I say she's correct. Is she correct? Yes. What? Yes. You want to give us another definition? She's perfect. Perfect. Facultative anaerobes can survive in both air and no air. Whether the air contains oxygen or not, it will still survive. Whether this air contains sulfur or contains ammonia. It's not air, it's oxygen. If you go and write air, I'm going to ask you that question. If you go and write air, because air contains many gases, that means it can be nitrogen. You're talking about oxygen now, not air. Then, facultative anaerobes. I'm particular about it, or facultative aerobes. Which of them do you like? Is it facultative aerobes or facultative anaerobes? Anaerobes. Define it. Define facultative anaerobes. What? Speak out, speak out. What? What happened to the bent horse? Yes, facultative anaerobes. Define it for us. Yes, you. Yes, you with the cap. Facultative anaerobes. Define it. Facultative anaerobes, strict anaerobes. Then we have strict aerobes. Then we also have what? Then we also have facultative aerobes. This one. Strict anaerobes. If oxygen is present, they will not be there. Oxygen must be absent. It's not air, it's oxygen. These strict aerobes, oxygen must be present. We also call it obligatory. Instead of strict, you can say obligatory. And I'm going to ask you, strict aerobes, obligatory aerobes, differently. So I will see whether you know it. Then strict anaerobes, strict obligatory anaerobes. Then facultative, you are doing the wrong meaning for me, because that's what I just told you. The assumption of this material assumes that you have done that assignment. If you have done that assignment, you will know the correct thing. That thing is not totally correct. The definition I just gave you now in that material is not totally correct. It's from the material. So that's why when they give you one assignment, go and do it very well. Then when you now sit inside the material, you now know how to now know that, no, this one. That's why I told you that these Enterobacteriaceae, they are intestinal in origin. They are from the intestine. And from the intestine, you go to use the water closet. Then you defecate. You defecate, not urination now. You defecate, excreta. Since they are intestinal in origin, they will come out with the excreta. We now also said that these same Enterobacteriaceae, they can be found in the environment. That means not intestinal in origin. We also say they can be free living, just stay on their own, in the environment. And they can from there enter water that you want to drink. Now you now have water. Who has water there? That's pure water that is there. Now you want to drink, you want to take it and test it in the lab. You now see E. coli in the water. Are you now going to drink the water? You will not drink the water because Enterobacteriaceae are intestinal in origin. Why didn't you assume that they are also free living and be drinking the water? And whether this water from this man's company is from free living, from that company, these are the part that didn't answer any question, is from polluted, from excreta. They didn't treat their water. Because you now see the Enterobacteriaceae can be free living. You now see it in your water, you now say, it's free living. It's from free living. It's from free living. You now drink the water. Whereas it's from the polluted place. It's from excreta. Inside the excreta, you have Vibrio cholerae, a pathogen, clear-cut pathogen. There's no how you take it. Before 24 hours, you'll be visiting the toilet. Before you go, you come back. Before you come back from the toilet, you run back. Before you come back from the toilet, you run back to the toilet. What happened is the water. But you saw E. coli in the water. You now assume that it's intestinal. You assume that it's free living. You get it now. The same thing I was telling you about facultative anaerobes. I gave you the assignment deliberately so that you can go and sit down with it. If I give you an assignment, go and sit down with it and look at different definitions. Because the way it is defined now, that means the same way we are going to define facultative aerobes then. They say they can survive in the presence of oxygen and absence, Abi. The two of them. But that's not the case. I taught you diauxic growth the other day. Is that not so? What is diauxic growth? I think I use somebody as an example. Okay, you are the one? Yeah. What is diauxic growth? It's going to come out. It's going to come out in your test too. What is diauxic growth? All right. Now, strict anaerobes, oxygen must be present before they can survive. But facultative anaerobes, they are aerobes. So you can see now, it didn't change. Facultative anaerobes, they are aerobes. But in the absence of oxygen, they will still survive. While facultative aerobes are anaerobes. It's opposite, but it can't move to the other side. So it's not just that facultative aerobes can survive in presence of oxygen, absence of oxygen. You now say, I got it, then the man wrong it for me. I will not take the primary one definition from you. It's the 300 level definition I'm going to be expecting. 300 level. After you've done this course, you're supposed to be a master bacteriologist. When you are past bacteriology, what remains? Anything bacterial, then you should call you and just call you and say, any question you want to ask you on bacteria, I should ask you. Abi? Is that not so? After you are taking this bacteriology, this is the study of bacteria now. After you have passed this course, you get A or B or C, or D or even E. Then any question on bacteria they ask you, you should know it. Any question. So that's what I'm telling you now. You just say, because I just said it in this book now, that this, and then you just take it like that. If you go and define what is defined here, that facultative anaerobes can survive in place of, no. Facultative aerobes, anaerobes, they are aerobes, but they can survive in the other one. Now, let's go. What I ended with are, some are free living, while others are opportunistic or primary pathogens. Primary pathogen means no story. That's why I give you an example. The primary pathogen. Some are opportunistic pathogen too. Classification. The family Enterobacteriaceae belongs to domain bacteria. They belong to domain bacteria. Phylum. Proteobacteria. Proteobacteria. Class, Gamma Proteobacteria. Gamma Proteobacteria. Order, Enterobacteriaceae. Enterobacteriaceae. Family Enterobacteriaceae. Enterobacteriaceae. Genus. What is the genus? They have different genus. So write genus, please. The family includes several important genera. The family includes several important genera, Genera, such as Escherichia, Come on, you underline it. Salmonella, Salmonella is somebody's name. Salmonella. Then Shigella. Shigella, can spell that. Underline them in each case. Shigella. Enterobacter. Serratia. 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 Proteus. Proteus. Yersinia. Yersinia. Citrobacter. Citrobacter. Providentia. CIA at the end. Providentia. CIA. The medically important genera and their characteristics are. The medically important genera and their characteristics are. Number 1, Escherichia coli. The medically important genera and their characteristics are. Number 1, Escherichia coli. Common intestinal commensal organism. Common intestinal commensal organism. Common intestinal commensal organism. It is an indicator of fecal contamination of water. It is an indicator of fecal contamination of water. Indicator of fecal contamination of water. When you get to 400 level, you understand this more. But if you catch it now, you just flow with it later. Some strains of E. coli cause urinary tract infections, UTIs, traveler's diarrhea. I remember we went to a conference somewhere at Delta State. That was in April this year. And as we went for that conference, please let's be quiet. Yes. Okay, infantile diarrhea. Septicemia. Septicemia and meningitis. Especially in neonates. Meningitis, especially in neonates. Meningitis, especially in neonates. Mr. 99, FFF. What do you mean by neonates? Mr. Man, Mr. Ankara, what do you mean by neonates? Yeah? Mr. Alfa, to go back to saying, what do you mean by neonates? What do you mean by neonates? Do you want to help them? Neonates. Neonates, newborn baby. Neonate. You've not heard of neonata. You've not heard of neonata, all of you that asked the question. Neonata. Therefore, neonata, neonates. That's a newborn baby. Number 2, Salmonella species. Salmonella species. Look at the species here, S-P-E-C-I-E-S. Salmonella species. When you write the name of organism, underline it too. You don't underline it, I may say, okay, instead of 1, give them half. Underline it. That's part of what we learn in bacteriology. So pay attention now. We get it now. So this, we don't give you, I'm explaining saying everything now. Exam now, you go and write Escherichia coli, this and that. You don't underline them. Is that you are breaking a rule, a scientific rule. What's the rule called? What? Biosurfactant. Binomial nomenclature. Thank you. And they have taught you that thing from SS1, Abi. You went to a good school. SS1, binomial nomenclature. It's not a play. It's real. Carolus what? Carolus lineals. Any scientific name you are writing, underline the genus name and the specific name separately. It's a rule on it. So even at the project level, some students are writing project because it's project title. Because the name is in the title, they will not capitalize it. They have broken the rule of binomial nomenclature. So you always say that they are there. When you've known this one, you know this one, you know this one. By the time you get to some point, when you everything just come out, where is this one from? The same thing, if you don't know it, you now come and you are putting all the capital. We don't look this one. Don't you know binomial from SS1? If you even do advanced and go to taxonomy, you still see binomial nomenclature there. So it looks, if you get it from SS1, you get to understand it. It's a rule on its own. You cannot say because it's tied to, you now break the rule. It's a rule. You get it now. Salmonella species, pathogenic to humans and animals. Pathogenic to humans and animals. Transmitted through contaminated food. Transmitted through contaminated food, water, or contact with carriers. The diseases they cause are typhoid fever, in bracket, Salmonella typhi, in bracket, typhoid fever, then in bracket, Salmonella typhi, Salmonella typhi. Then the next one, paratyphoid fever. Paratyphoid fever. In bracket, Salmonella paratyphi. ABO. Capital A, comma, capital B, comma, capital O, comma. So it can be Salmonella paratyphi A, Salmonella paratyphi B, Salmonella paratyphi O. Then the next one under the disease is food poisoning. Food poisoning, also called gastroenteritis. 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 Food poisoning. In bracket, Salmonella enteritidis. Oh, let me confirm you. Yeah, enteritidis, just the way I call it. Comma, Salmonella typhimurium. Salmonella typhimurium. typhimurium. Salmonella typhimurium. Number 3, Shigella species. Shigella are non-motile. Shigella are non-motile, non-lactose fermenters. Shigella are non-motile, non-lactose fermenters. They cause bacillary dysentery. They cause bacillary dysentery. Bacillary dysentery. In brackets, shigellosis. So the dysentery are caused by different organisms. You understand? Shigellosis. Characterized by diarrhea with mucus and blood. Characterized by diarrhea with mucus and blood. Characterized by diarrhea with mucus and blood. You can see now, the Shigella diarrhea has mucus in it, blood is there. Why is it so? Because the organism is eating part of the walls. So when something is coming out, it will have blood and mucus in it. And then there's some kind of decomposition taking place. Transmission is fecal oral route. Transmission is fecal oral route. Have you heard about that before? Fecal oral route. The person drinking pure water, where are you? The person drinking pure water there, where are you? You are the one? Are you the one drinking pure water? You are the one? You. What do you mean by fecal oral route? What do you mean by fecal oral route? Here. Fecal oral route. What do you mean by fecal oral route? Fecal oral route. Have you heard about fecal oral route before? Yes. So what is fecal oral route? You are drinking pure water. I've explained it before already. Do you want to help them? You want to tell them? Fecal oral route. What? Through mouth. What is fecal? So what is oral? So what is fecal oral route? Eh? I want one person to talk now. One person that is confident. Confident. God. Are you raising your hand? One person that is confident. What is fecal oral route?
Like you explained that maybe you see one bacteria like
Speak out. Continue. Yeah, you're on the right path.
like you explained then maybe you see one bacteria that
One bacteria.
no
One
like,
bacteria.
Like, if you see a bacteria
If I see a bacteria,
if you see bacteria in maybe in a food.
if I see it
Yeah, in a food. Now
in a food, I mean in food.
in food. And it causes and it causes transmission of through feces. Maybe
Fecal oral route. What is fecal? Feces, excreta. What is oral? Mouth. Simple. Fecal oral route. Just explain it. How does what? Contaminated food. Food contaminated with feces. That's it. So I said transmission of fecal oral route through contaminated food or water. Through contaminated food or water. Through contaminated food or water. That is why we said, from primary school, we said when you defecate or excrete, what should you do with your hand? Use it to wash soap and water. So that the hand you use to clean up, you will not go and use it to eat rice and spaghetti. You know, some people will say they don't like using spoon again. They like using hands. Some people will say that. In some cultures, like in India, they use hand. Rice, whatever, you see them eating with hand, you'll be doing like this. Hey, all of them, whether professor, whether whoever, big man, using hand. But if you're a visitor, they will give you a spoon. They say, don't worry. They know that you like spoon, take spoon. But spoon has its own problem too. Because you don't know who used the spoon before you came to use it. That's why it's best you go with your spoon in your bag. You get it now? It's best to go with your spoon in your bag. If the washing does not remove organism from the spoon, as a microbiologist, you should know that, except sterilization. So you go to canteen, you go to everywhere, you're using spoon. So there's microflora movement. So talking about fecal oral route, it's usually in places where you don't have sufficient water, where there's not enough water. Most of these problems are linked with water. That's why when you come to water microbiology, we sit down on it and talk about it and give you different scenarios. Most of them are linked with water. Insufficient water leads to transmission by fecal oral route. In a place where you don't have water closet, no water closet, the child defecates on the floor in the kitchen. The child excretes on the floor, right in the kitchen. It also happens in poor communities. No Pampers is used. In rich communities, there's carton and carton of Pampers. Use one in the morning, use one in the afternoon, use one in the night. Use one to sleep, 4 per day. But in poor communities, they will use cloth Pampers. They will sew the cloth and tie it, and use it. When they will not wash it, and the mother will not wash it, the mother will give it to another person, a young chap like that, older than the initial one, maybe 10 years old. That one does not even know anything that's fecal oral route. Even the child, before they clean this thing, the child himself will have touched the excreta. And children, even adults, go and watch children when you get home, in your place where you are living at Tokyo or anywhere. Just sit down, or any shop inside the school. After this lecture, just go there and be watching children. Anything, they put it in their mouth. So as they excrete, they want to see how this excreta is sweet, or they put it in their mouth. Other children, the same thing. During washing, during everything, transmission is taking place. Let's go to number 4, Klebsiella species. Klebsiella species, non-motile encapsulated rods. Non-motile encapsulated rods found in soil, water, and the human intestine. They are opportunistic pathogens causing pneumonia. They are opportunistic pathogens causing pneumonia, especially in alcoholics. Causing pneumonia, especially in alcoholics. So you can see why I said that line, that red line, leave the space. You now be wondering, why is it causing pneumonia in alcoholics? Go and read about it, then write it on the side. But this one, add it to your assignment. Why do they cause pneumonia, especially in alcoholics? Comma, UTIs, septicemia, UTI, urinary tract infections. UTI, septicemia. Septicemia, it's from the word sepsis. Sepsis. It's from the word sepsis. It's now called sepsis. Then wound infections. Wound infections. Number 5, Enterobacter species. Number 5, Enterobacter species. Enterobacter species. They are motile opportunistic pathogens. Sorry, they are motile opportunistic organisms. Yeah, correct. Pathogens, correct. They are motile opportunistic pathogens or opportunistic organisms. They are involved in hospital-acquired infections. They are involved in hospital-acquired infections. They are involved in hospital-acquired infections. What is another name for hospital-acquired infections? What? Cocosomia. Nosocomial. So nosocomial infections are hospital-acquired infections. What about community-acquired infections? What about community-acquired infections? That one is called what? Hospital-acquired infections are called nosocomial infections. What about community-acquired infections? Yeah? It's still nosocomial infections. Because some people will not go, they will have infection, they will stay in their house. Though. You understand? Some people have infection, they will not go to hospital. So it's not only hospital that they acquire infection from. Somebody can have infection and stay in his house. They have tuberculosis, stay in his room, and you go to greet the person and he's coughing. Say sorry. The spoon the person used, go and take it, use it. The droplets, you take it, you inhale it, everything, then you go. It's in the community, it's not in the hospital. So whether somebody is in the hospital or is at home and sick, you should pick race. And if you don't pick race, you go there, be careful of what you touch. Whether you touch anything or not, leave the place and go and wash your hand with soap and water. And if possible, don't eat with your hand throughout that day. So you eat with your fork. And if possible, also go with a nose mask. So let's go on now. Hospital-acquired infections such as respiratory tract infections, wound infections, UTIs. Enterobacter has been referred to as the twin. Twin of what? Twin of E. coli. Enterobacter has been referred to as the twin of E. coli. Full stop. Part of your assignment now. Why is it referred to as twin of E. coli? To add it to your assignment. Why is it referred to as the twin of E. coli? Number 6, Serratia. Serratia marcescens. Serratia marcescens. That spelling is not correct. Marcescens here. Serratia marcescens. It produces a characteristic red pigment. It produces a characteristic red pigment. It produces a characteristic red pigment. You see, that's why this thing is very simple. If you understand the theory really very well, by the time you now go to the practical, you now go to project. In fact, it's very interesting. I find it very easy. Maybe because I read. If you read, you find it very, very easy. It's only Serratia that produces that. When you grow it on nutrient agar, it will give you characteristic red pigment. But that does not mean that it's only Serratia, but at least you have moved in one direction. Then you cannot carry out some other tests specific for Serratia. So you cannot do gram staining and see the shape is still raw. Then you can do some specific tests for Serratia, and then you can say it's Serratia. But if you don't know that it has red pigment, you cannot know that it's supposed to do some specific tests for Serratia. Produce characteristic red pigments due to the possession of, are we safe? Due to the possession of proteus. Proteus. Due to the possession of proteus. Serratia is an opportunistic pathogen causing pneumonia. Serratia is an opportunistic pathogen causing pneumonia, endocarditis. It causes pneumonia, endocarditis, urinary tract infections. Urinary tract infections. The next one is Proteus species. The next one is Proteus species. Proteus. Proteus. Proteus species. They are highly motile. Wow. As I'm reading it, I'm just seeing it on the plate. They are highly motile. So with that, let me just ask this question now. Let's say this is the plate. Abi? This is your petri dish. I mentioned this the other time. This is your petri dish. And then you strict it, you do pour plate with Proteus, and it's highly motile. So what's going to happen on the plate? What? I mentioned it clearly in the last class or so. Swarming. It's going to be swarming. That's what it's called too numerous to be counted. Because it grows, it's motile. It grows very, very rapidly. It can move very rapidly. And if it's moving, it's not like human being that if it's moving, it will just move. It's moving because of its growth. We move because of locomotion. It is moving based on its growth, and it's rapid. So it's within, normally E. coli in 18 to 24 hours, you just see it on the plate. Colonies. This one will swarm the plate. As if you flooded the plate with, you just pour some water on the plate. It will flood the plate. But you see the whitish or characteristic color that, yeah, this is organism. It will flood the plate. So you cannot count and pinpoint because it's motile. So the colonies will not be distinct. They'll be interconnected, and then they'll form like a single, like when you pour water on the something. So I said it's highly motile. It has swarming motility on agar. It has swarming motility. It has swarming motility on agar. See, this is just the starting point now. Somebody have told you about, the other person taught you what? What did Mr. Emmanuel teach you last week and yesterday? Classification. We have not started yet. This is the first one. This is the first family. Other people will come with different families. If you don't grab the Enterobacteriaceae family, when they come with another family, and they come with another family, you will now be looking, is it in Kende family or Idowu family? Yeah. Or is it in Alaba family? Because you didn't get that one. By the time the next one they come to arrange, you'll be asking that who is the first one? Is it Taye or Kende? Then you start asking yourself that Idowu and Alaba, who is the senior? You understand? But if you understand it properly from Taye Kende level, you've known that one. Then they come with Idowu, you know that Idowu is this one that follow them. Then after that Alaba, you know that Alaba that follow them. If you don't do that, you now wait, everything, you just pile it up. By then you look at is this Enterobacteriaceae or another chlamydia or mycoplasma? They produce urease. They produce urease. 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 They produce urease, which splits urea into ammonia. They produce urease, which splits urea into ammonia. Raising the pH of urine. Raising the pH of urine. Raising the pH of urine. They cause wound infections. They cause wound infections. Please, let's be quiet, please. They cause wound infections. UTIs. They cause wound infections, UTI. Kidney stones. Kidney stones. Due to alkaline urine. Due to alkaline urine. Number 8, Yersinia pestis. Pestis. Yersinia, Y-E-R-S-I-N-I-A P-E-S-T-I-S. Pestis. This is the constitutive agent of plague. This is the constitutive agent of plague. P-L-A-G-U-E. This is the constitutive agent of plague. P-L-A-G-U-E. Is she okay? Are you okay? Are we in the month of Ramadan? What? Not class for what? For so when you exam, what you should be doing? Concerning agent of plague, in bracket, bubonic. Bubonic, B-U-B-O-N-I-C. Bubonic plague, pneumonic plague, septicemic plague. So in bracket, bubonic, pneumonic, septicemic. So they cause plague. So the plague can be bubonic, it can be pneumonic, it can be septicemic. Transmitted by flea bites. Transmitted by flea bites. F-L-E-A. Transmitted by flea bites. F-L-E-A. Or contact with infected rodents. Or contact with infected rodents. Biochemical reactions heading. Biochemical reaction used for identification. Biochemical reaction used for identification of Enterobacteriaceae. Biochemical reactions used for identification of Enterobacteriaceae. Actually, this is like a table. Is that okay? So in the table, let me just tell you the heading. Then you decide. We'll agree on which one we carry the largest part. On the table, there are 3 parts: test, principle or reaction, and then example of positive results. Test, principle or reaction, example of positive results. So the test should not be too big. Principle stroke reaction should be a little bigger. Then example of positive result should be just big as the test. So test and principle of example of positive result should be the same largeness, but the principle of reaction should be larger. That's all. Test number one test. Indole test. Indole test. INDOLE. Indole test. The principle of the reaction is that tryptophan is converted to indole. Tryptophan, product sign, indole. Tryptophan, then product sign, indole. Example of positive results: E. coli, positive. E. coli, positive. Number 2, methyl red. Methyl red. Methyl red. What is the acronym for methyl red? MR, Abi? MR in brackets. Methyl red test. Principle: detects acid from glucose fermentation. To detect acid from glucose fermentation. Detects acid from glucose fermentation. E. coli positive. Number 3, Voges proscure. Voges proscure. Voges proscure. Voges, V-O-G-E-S. Proscure. P-R-O. P-R-O-S. K-A-U-E-R. K-A-U-E-R. That's VP, Abi. Voges proscure. That's VP. Detect acetoin. Acetoin. A-C-E-T-O-I-N. Detect acetoin from glucose fermentation. Detect acetoin from glucose fermentation. Example of positive results: Enterobacter positive. Enterobacter positive. Number 4, citrate utilization. Citrate utilization. Principle: uses citrate as carbon source. 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 Klebsiella. That's example of positive result: Klebsiella, Enterobacter positive. Klebsiella, Enterobacter positive. Urease test. Urease. U-R-E-A-S-E. Urease test. Splits urea into ammonia and CO2. Please, let's be quiet. Let's be quiet. We'll listen. We'll get it if you are quiet. Oyinbo pepper. See, Oyinbo is discussing there. Oyinbo pepper. Please, let's be quiet. Is that okay? Urease test. Splits urea into ammonia and CO2. Splits urea into ammonia and CO2. The organism is Proteus, Klebsiella, positive. Proteus, Klebsiella, positive. Then the next one is triple sugar iron agar. Triple sugar iron agar. Triple sugar iron agar. In bracket, TSI test. TSI, triple sugar iron. Triple sugar iron test. TSI. It detects glucose. Detects glucose, lactose, sucrose fermentation. Detects glucose, lactose, sucrose fermentation. H2S, gas production. So that's what that test is used for. It's just one single test. You prepare it's an agar. So prepare the agar in the autoclave. Prepare it on inside test tube or MacConkey bottle. Then you make sure it is slanted inside the bottle. Then you now streak the organism on the agar. Then incubate it. By the following day, the color will have changed. If it's this color, if it's Oyinbo color, it is, what's that guy's name? Jamil. If it's Oyinbo color, it's Jamil. If it's wearing white cap, it's this guy. If he's using glasses, it's this person. That's all. You understand? So if it's also color, it is this organism. If it's also color, it's positive for this, or negative for this. So the organism is used for Salmonella produces hydrogen sulfide. Salmonella, example, Salmonella on the organism side. Salmonella produces H2S. H2S. So you are going to have, in that agar, you are going to have black bugs. Black, B-U-T-T. So this is the test tube, and this TSI you are prepared. So this place will be black if it is Salmonella. If it's another thing, it will have different color. Motility test. Motility test. Please, let's keep quiet. I don't want this noise. Let's be absolutely quiet. Motility test detects movement. Detects movement. In the third section, Proteus, is it motile or non-motile? Proteus, motile. Klebsiella, non-motile. So if the class rep brings an organism to the class and he said, the lecturer said this organism is motile or is Proteus, you don't just take the organism and start using it to do your research. You try to confirm the organism. You understand? Or I travel to America or to Germany or to Canada. And I now send, you now ask me that, please, sir, help us get that organism, Proteus. And I send it to you. That this Proteus, you've checked it in the best place in USA. As it comes, just take it and start using it. Say, it's Proteus. So God has sent it. No. You check for motility. If you streak it on the plate, you swam the plate, then you say, wow, this is Proteus. You get it now? You get it? Let's move to the next thing we're doing. That table is done. Pathogenesis and virulence. Subheading. Please, let's be quiet. Yes. Pathogenesis and virulence factors. Virulence. Pathogenesis and virulence factors. Pathogenesis and virulence factors. Pathogenesis and virulence factors. Many Enterobacteriaceae species possess virulence factors such as but before we go on about virulence, what are virulence factors? That causes disease. That's the what makes the organism to cause disease, what we call virulence factor. All right, let's go on. So you can write that somewhere. Because I may ask you, what are virulence? Maybe all the test will just be question. What is it? What is it? What is this? 10 of it like that. Just normal question 1. Only one question I'm going to set. Everybody is going to set one question like that, or two questions. So what is A, virulence factor? B, this. C, this. D, this. A, this. What is generation time? What is doubling time? What is this? The class rep gave you CFU per something of these diseases. What is this? Using formula, calculate the that kind of question. So after you to go at this formula now, you didn't use it. If you like, don't use it. Don't get it. When you get it, you see, formula. This man told us. You know me, I'm very simple. I will tell you the question straight and direct. The question is coming out. This one is coming out. Everything I'm saying will come out. When I'm setting it, I will set it. Pathogenesis and virulence factors. One thing I know, I've taught you bacterial growth. There's no how you will not draw the growth curve now. Abba. You must, it's a must now, Abi. You must draw the growth curve. It's a must. What's this one? For those who don't come to class, don't tell them, well, I know you have more than 300, but here now we're having about 100 or so. Many Enterobacteriaceae species possess virulence factors such as endotoxin. Endotoxin. That's the first virulent factor. Endotoxin. Lipopolysaccharide. Lipopolysaccharide. It causes fever. It causes fever. It causes shock. It causes fever, shock. Exotoxins. E.g., enterotoxins in E. coli. Exotoxins, e.g., enterotoxins in E. coli. The next one is capsules. Capsules. C-A-P-S-U-L-E-S. Capsule. Bacteria have capsules. Protect against phagocytosis. The bacterial capsule protects against phagocytosis. So when a bacteria have capsule and it infects somebody, and the person go to chemist or go to hospital and they give the person drug that is going to attack capsule. But the organism already have, how will I put that example? Let me look at the good example again. So capsule protects against phagocytosis. It protects against breakdown so that the organism will not break down by the action of a drug or antibiotic. So even when you use the drug, That's why they do what is called antibiotic sensitivity test, to know the best drug. And that's why they are saying that stop abusing drug. When you have infection or anything, go to doctor, medical personnel. Don't come to Dr. Adetutu or any other person on radio or post office shouting, Staphylococcus, Staphylo whatever, that you do not know. Go to a proper medical doctor. When you go to the doctor the first time, he give you a drug. It didn't work. Go back to the doctor. Doctor, I'm still feeling this way. Then the doctor will now raise the tempo. Don't go to another doctor again. Then the next one is Fimbriae. 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 In brackets, pili, P-I-L-I. This one aids in adhesion to host cells. Aids in adhesion to host cells. Aid in adhesion to host cells. Aid in adhesion to host cells. I know some of you live in Lagos. I don't know how Lagos is now, but those days, you must run and pursue the vehicle. You still pursue vehicle now? Yes. You still pursue vehicle in Lagos? I've been in London for some time now, so I don't know. And you still pursue vehicle up till now? So when you pursue the vehicle now, so what do? You now hold the vehicle, and put your leg as it's going, and you start the balance. So that's how the Fimbriae is too. That's how the Fimbriae is. When the organism wants to infect the individual, it will attach using the Fimbriae to hold, so that the person will try to, the system will try to log off the organism. But the organism will use something to attach to the host cell. As attach, then to attach, then to balance, then to settle. Just like when you enter bus in Lagos. Those days, Molue. You didn't enter Molue. There's no Molue again. So in Molue, it's a big bus like that. You run, then you hold it, you jump, hold it, put your leg and hold. As it's going, you now be climbing, be entering the vehicle. So as it's going again, you look for seat, you move inside and sit down. Sorry. So the same thing with the Fimbriae or pillar. It aids in adhesion to host cells. Aid in adhesion to host cells. Aid in adhesion to host cells. The next one, plasmids. 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 They carry genes for antibiotic resistance and toxin production. They carry genes for antibiotic resistance and toxin production. Now, let's look at the epidemiological importance of Enterobacteriaceae. Epidemiological importance of Enterobacteriaceae. Number one, they are common agents of nosocomial. They are common agents of nosocomial. They are common agents of nosocomial. I think you now know that nosocomial infections are hospital acquired and non-hospital acquired, community acquired. It's not just because even here it's saying hospital acquired infection. But somebody may be sick and not go to hospital. So if you go and visit the person, you can acquire common agents of nosocomial infections. Number two, cause outbreaks. Cause disease outbreaks. Cause disease outbreaks through contaminated food, water, and medical devices. Through contaminated food, water, and medical devices. Number three, increasing concern due to antimicrobial resistance, AMR. Laboratory diagnosis of Enterobacteriaceae. That's the next subheading. Laboratory diagnosis of Enterobacteriaceae. How do we determine or isolate Enterobacteriaceae in the laboratory? Laboratory diagnosis of Enterobacteriaceae. Number one, specimen collection. Number one, specimen collection. If you go to the lab, if you go to your microbiology lab and you want, they brought water sample and they used to bring every time. And the practical you are going to do, I will design a practical that is going to involve you collect water, you analyze the water for E. coli. Number one, specimen collection. So what is the specimen going to be? It can be stool, urine, blood, wound swabs, water. Stool, urine, blood, wound swabs, water. Number two, culture. On selective stroke differential media such as A. MacConkey Agar. MacConkey, M-A, small c, capital C-O-N-K-E-Y. MacConkey Agar. In bracket, lactose fermenters appear pink. On MacConkey Agar, lactose fermenters will appear pink. Then B, EMB Agar. EMB. EMB is eosine methylene blue. Eosine, E-O-S-I-N-E. Methylene blue. EMB Agar. In bracket, metallic sheen for E. coli. Metallic sheen. Sheen, S-H-E-E-N. Greenish metallic sheen. Greenish. But here I say metallic sheen, but it's greenish metallic sheen. For E. coli. Salmonella, Shigella, Agar. That's the C. We're talking about the culture now. Salmonella, Shigella, Agar. For Salmonella and what? Shigella. So you get the agar. It's called Salmonella, Shigella, Agar. Prepare it. Then, if Salmonella is in the sample, you can do pop plate, you can do any kind of stick plate, you can do spread plate. If Salmonella is there, it will have a characteristic color and growth. If Shigella is there, it will have a characteristic color and growth. So that side by your notes, what is going to be the characteristic color and growth? You should go and read more about that and write it by the side. That Salmonella will appear so on Salmonella, Shigella, Agar. Shigella will appear so this way. Number 3, gram stain. Gram stain. How is it going to be on the gram stain? How is it going to be on the gram stain? Gram negative. Why are you changing this like that? When I say positive, when I say positive, is the stain, is it going to change on the... Yes, gram stain for Enterobacteriaceae. You standing, that lady. What is it? Is it going to be gram positive or negative? You. Yes. The gram stain for Enterobacteriaceae, is it going to be positive or negative?
Negative.
This is your first time of coming to the class? Eh? No. You live in Okyodo or on campus? Eh? What? Off campus. Okay, you are forgiven. It's the transport that caused the problem. It's the transport. Time that you're supposed to read, you use it for transport, queue for transport. So gram stain, gram negative. All these things are established already since the beginning. Gram negative what? Gram-negative what? Eh? Gram-negative rods. Gram stain, gram-negative rods. Biochemical tests for species. Number 4, biochemical tests. Colon. For species identification, there are different tests. Each species have different tests. For species identification. So there are different tests that you do to identify the species. Number 5, serological tests. Serological tests. Salmonella and Shigella typing. Hey, please, let's be quiet. Salmonella and Shigella typing. Antibiotic susceptibility testing. Antibiotic susceptibility testing is used to guide treatment. I've said that before. Abi? Treatment and control. Treatment and control. Subheading. Antibiotic therapy. Number 1, antibiotic therapy. Based on susceptibility results. Antibiotic therapy based on susceptibility results. Then rehydration therapy. Number 2, rehydration.
Based on susceptibility
results. Rehydration therapy. In diarrheal diseases, you have to rehydrate, especially in children. C, prevention. Prevention. Proper sanitation and hygiene. Proper what? Sanitation and hygiene. Proper. That's why anytime I come to class, you open the window. Before somebody has something now, the window is not open. You have to be spreading all over the class. Sanitation. Sweep the floor. Disinfect. Mop the floor with disinfectant from time to time. Especially if you move into a new building. You should mop it, sweep it, and mop it with disinfectant. Not just wash it with water and this and move in. When you go on break, you come back, you do the same thing. Wake up in the morning, open the window, let UV light move in to kill some of the antibacterial and some of the bacteria in the space. The next one, safe food and water practices. Safe food and water practices. Infection control in hospitals. Number 3. Infection control in hospitals. Then number 4. Rational or rational antibiotic use. Rational or rational antibiotic use to reduce resistance. If you are just using antibiotics by yourself, you are going to be increasing resistance. That's why I said the doctor will not give you the best the first time. He will not give you the highest, or not the best, the highest the first time. He will give you one he thinks it will take care of what you are dealing with. So you don't run away from the doctor. Go back if it's not done. Then he will increase it. You go to another person now, they just give it, scatter the treatment procedure. Any question? Any question on Enterobacteriaceae or anything I've taught you? Any question? What? Rehydration therapy, yeah. What? What do you mean by rehydration therapy? Madam, pure water people there. What is rehydration therapy? Madam Onifila, Dudu. What is rehydration therapy? Water. But if somebody has diarrhea, is he drinking water? If somebody is having diarrhea, is it water he's going to be drinking? Somebody is having diarrhea. You don't have diarrhea now, you're drinking water. That's still rehydration. But the baby or you're having diarrhea. What do you take? What? Yeah? ORS. So you take salt sugar solution. You take salt sugar solution. So that's fine. Any other question before I go? Any other question? So somebody else will continue next week.
Thank you.
Have a good one.